Okay, now for the novel part of the meeting. The plan, as I think you heard from Adam, is questions really have to be sent in so they can be read here. We won't have microphones, and if they're too long, I won't read them. So keep them to a sentence. So we have a few questions here for the, should I be mentioning who the questions come from? Like Julio or whatever? So one of the questions is, how does the target organ genetic evolution? So who would like to answer that question? And Robert, we're talking about target organ influence, obviously.
Right. So in uveal melanoma, the primary metastatic site is the liver. We don't understand that at all. You know, we've tried to look at chemokine receptors, for example, chemokine ligands, chemokine receptors, and it doesn't really make sense because there was this story about this with breast cancer, for example, metastasizing to the lung. So it's something we struggle with all the time, something we need to understand.
Adam, Jennifer, can he talk? Okay, go ahead.
So we have exome sequenced and genome sequenced the few metastatic tumors that are available, and there's some data now in the literature as well. And there aren't new drivers, there aren't new changes in the metastases. So that's.
But following up, I mean, in fact, you know better. I mean, BAP has
the BAP mutation.
Mutations have effects on other organs besides uveal melanoma.
Yes, so there's a familial form of cancer where there are BAP mutations, familial BAP mutations, which isn't, like mesothelioma, for example, and a small proportion of cutaneous melanomas and renal cancer. So BAP is mutated in other cancers, but not to the same degree as in metastatic uveal melanomas. But BAP is, you know, all the mutations we see in the primary tumor, we see in the metastasis. But in terms of what's going on in the target organ, it's something we really need to understand. We need to talk.
Which has some of the same mutations, you're saying?
We definitely need to talk.
But I mean, we know that you can have same mutations in different organs with a very different effect, obviously. But I think your point is we don't really understand mechanistically how that occurs. Which actually leads to this other question. Are there host genetic polymorphisms that affect the rate of relapse? Robert, you should be able to answer that.
Lisa, I'd say the answer to that definitely is yes. Certainly, I think, like for instance, the variant I showed is the kind of variant that probably, we didn't specifically look at rate of relapse, but probably something like that is associated. And going back to the first question, I think such inherited polymorphisms could affect either the biology of the tumor itself, or could also be affecting the biology of either the site relapse occurs, the site metastasis occurs, and enabling that and allowing that to happen more readily. So there are a lot of different possibilities in terms of the mechanism there.
Well, here's another question which is both important and I would say philosophical. Because the question is, how precise does precision medicine need to be? Or, in other words, how many angels can dance on the head of a pin? Who wants to answer that? Judy, you should be able to answer that.
We actually had a good discussion on this in the Tisch meeting on Monday. And so the question is that instead of having kind of a standard kind of screening, everyone should get screened at 50. But can we modulate this a little bit? If you're like in the bottom 10th percentile or the top 10th percentile for polygenic risk scores, maybe we can shave it a little bit so that instead of just being 50, it's 52, 55, or earlier, 45. And so in the case of just simply modulating by a few years when you're going to do the screening test, I don't think you need to be that precise, but I think you should know that you're in the top 2% of polygenic cancer risk score, so it's time to do it earlier. For other things in terms of, so that's kind of the early diagnosis issue or prevention. In terms of the interception thing, I think the need for precision is going to be a tad higher.
No, actually, I think that's a very good response. I think you're right. Early on, and actually, you can also, can be age-related. Children have fewer mutations, which are picked up over time. So in fact, maybe age can determine the degree of precision that you can have as you just pick up random, maybe they're not random, maybe in fact calling them random may in fact point out some of the ignorance that we have about it, but you're probably right. Many of the mutations are going to be, we'll call them passenger, though maybe in time passengers will be modifiers that we don't know about today.
Can I comment on something, one point on the precision medicine? So currently, in the genomics landscape, targeted therapies don't really benefit a lot of patients. So this is one of the most disappointing, I think, aspects of precision medicine, at least with respect to cancer. And moreover, as someone mentioned about the organ side, targeted therapy some of the targeted therapies can work in specific organ, but they are complete failure in other organs. So we need to understand how to overcome and how to unleash the tumor to respond to different targeted therapies. So there is sometimes initial response, some response, but very quickly in different sites, the tumor somehow manage to overcome the initial response. So I think this is something that, so I will say that we need at least to be more, that precision medicine needs to improve chemo and one size fits all. Once we can manage to do it for all patients, it would be great, but I think we are still far.
Okay, so we do have some more questions, actually one for Gustavo. And it was, what are the benefits of the nano DLD platform over other liquid biopsy techniques?
Nano DLD is, it can separate different things such as viruses, DNA, but we are focusing on exosomes. We think that exosomes have, while DNA liquid biopsies are having a lot of scrutinizing, and I think that they are delivering reasonably good results. I think exosomes have a lot to offer, and because of lack of technology, we haven't seen enough good results with respect to exosomes. But it is known that it brings the messages that every tumor cell is sending or shedding out of the cell. So therefore, it is very reasonable that it's going to be a good instrument, exosomes, for early detection. Nano-DLD does some things that other technologies cannot do, like separating in a very clear range of sizes of vesicles, and we believe that that could be an advantage in being more precise in the diagnosis.
Right. And much more information in them, obviously.
Yeah.
So there's a question here from Ramon, and since he's my boss now, I better take it. It's for Anne, and it says, BAP1 is a dominant metastasis suppressor in uveal melanoma, and why is it not so simple in terms of how it functions in other cancers? Presuming that dominant suppressors is a simple way of understanding its effect in uveal melanoma. Or in breast cancer.
So I'm not quite sure I understand the question, but yeah.
You want to modify that?
Great model. It's a real human disease where you have a real genetic driver that can distinguish low risk versus high risk, predicting metastasis. My question is basically why in other solid tumors don't we have a similar clear signal like this? And I just thought maybe you'd thought about that and could sort of pontificate a bit.
You mean
about
why aren't they as clear cut?
Yeah, they're not, it's very, in most cases, for instance, in colon cancer, metastatic colon cancer, there's no clear genetic driver that's, distinct in the metastatic disease versus.
Okay, because I thought you were, because you can find BAP mutations in other cancers, and we have less a precise understanding of their effect.
Oh, that's a hard question. I mean, partly I think, uveal so the question is, why is BAP1, the effect of BAP1 in uveal melanoma so clear cut that mutations lead to metastasis, but why in other tumors, are there not as clear cut effects? Other genes. I think part of it is the heterogeneity. So in other tumors, they're often more heterogeneous than we realize. Breast cancers, there's multiple different cancer types, lobular, ductal, etc. Colorectal, I mean, we have the mismatch repair mutations in a subset. So I think, to me, I think about pediatric eye cancer, retinoblastoma, and that is very, very clear cut. And it's interesting that in a second cancer in the eye, which is in the adult eye, there's still some more clear cut answers than we usually see. Having said that, I mean, as I said just now, BAP1 is mutated in renal cancer. There can be germline mutations, so there are similar effects there. I mean, there's all kinds of other interesting questions as well. Why BAP1 in uveal melanoma and renal versus BRCA1 and 2 in breast and ovarian? Something we really need to understand.
I kick it back to Ramon. What's your answer?
It's a pity that it's not so simple.
That's not a great answer.
I could have said that.
I think the possibility is, as a clinician, that you have a phenotype, a very rare phenotype, that happened to have a single genetic driver that you were able to split based on the presence or absence of BAP1. When we look at diseases like prostate cancer or breast cancer, we classify them as a single tumor because they start from a single anatomic organ, and obviously that's wrong. And clinicians know that phenotypically all prostate cancers are not the same. So despite some of the data you saw, I can walk into a room and tell if a patient's going to do well or badly based on very cheap clinical biomarkers. And this is the key difference. I think uveal melanoma is something extremely unique and rare, and we were fortunate that you found a specific driver. I think that's probably a big part of this, Ramon, to answer your question, and you brought it up. The word is heterogeneity. The big complex diseases are extremely heterogeneous, and what we can't do easily is phenotype them.
I think what you said about prostate cancer is very interesting. I hadn't heard that, because obviously aggressive prostate cancer that kills patients is not that common, and we need biomarkers. And if we can use your eye and your insight, that's very, very interesting. I'd be interested to know what biomarkers you actually see, or what histological changes you see.
We still use a logic scoring system that forms almost every.
Like
Are you
Gleason
are you
score.
blaming Carlos or what?
I mean, yeah.
Even for the PSA?
Gleason. Yeah.
I know.
So the question is, if you do transcriptomic analysis, does it perform better? So in most cancers, transcriptomic analyses do perform very well, and 1 in 8 breast cancers only looks like they're going to kill the patient from transcriptomic.
But what's the test? Are we hypothesis testing, or are we predicting? With predicting, more data is better.
So, Judy, we have a question for you. Have any recommendations been made to clinicians regarding cancer screening based on BioMe data?
Yes, we've actually done it. And again, we'll actually have the folks that did it, Nora and Emer, talk about it. They just published a paper on Steele syndrome. And so what you find is Steele syndrome is a defect in one of the collagen genes, and basically identified by short stature Puerto Ricans. And basically, some of them are being treated inappropriately in rheumatology clinics. Some of them are being treated with like very potent anti-inflammatory agents. And so basically, this was first out of the gate. If you're a short Puerto Rican, you're more likely to carry this mutation, the collagen gene, and you want to make an affirmative diagnosis so that inappropriate therapies are not given. Not in cancer yet, though. So I don't know if you want to add to that, but that's basically kind of, and we just met with Glenn Martin about like these ACMG things have flipped the ethical paradigm, and so now it's like, how dare you give results back to patients, and then the paradigm is going to get flipped to how dare you not give results back to patients.
You know, had been mentioning, besides the Lynch syndrome, there's a large database being developed in children by a guy named Tabori up in Toronto, where they're coming up with some amazing data. There probably are many more mutations. Again, these are found in children with very high mutation rates, like 1,000 mutations, much higher than Lynch syndrome, as they delve into these databases. So I think we're just at the beginning of this area.
It's the beginning of a tsunami.
A tsunami, yeah. Am I allowed to, Adam, Jennifer, there's somebody raising their hand here? It better be a great question because you're out of turn, you know.
Yeah, I just asked for the
And you have 1 minute left.
Okay, I just asked for the prostate cancer for the GWAS analysis. Do you find any markers that related with different ethnic groups?
So that's definitely something we're interested in looking at this point, just because you really need cohorts that have long follow-up time to be able to do the analysis the way we did it. To date, I've only looked in these 2 cohorts from Sweden, but certainly looking at other patient populations, especially here at Mount Sinai, is something we plan to do.
Okay, I think we better stop here because I see the clock is counting down. We have 20 seconds left before we stop, so I think so let me use the 20 seconds to thank everyone. I think this was a terrific session. Thank Adam, Jennifer. I think this was a great start for the day, and we'll get ready for the second session.